Well, first I just want to thank John Stone and Jim Phillips and the organizer of this conference for inviting me to present my data. I'm going to actually be talking about something slightly different than the rest of this conference. Instead of doing, computations on individual molecules and within individual molecules on a quantum scale and molecular dynamics scale, my lab is more interested in systems of many, many molecules such that we can simulate an entire cell. And so instead of PME calculations, we're focused on diffusion and reaction, stochastic simulation of biochemical reactions. And so we're predominantly interested in a dynamic model of an entire cell under natural conditions. Now this is not yet achievable, but I'm going to show you some results that suggest that we're on the right track. So the overview of my talk, part one is I'm going to discuss a very common algorithm for a stochastic simulation of biochemical reactions under a well-mixed environment. And what I mean by well-mixed is that every reaction can feasibly take place in any portion of your reaction space. It's all equal. Then in part two, I'll talk about more of a spatially resolved stochastic simulation which we've developed, which allow us to sort of move away from the well-mixed scenario and into the more realistic scenario. And then finally, I'm going to show you a short simulation as an application of what we've been doing. And so I apologize if this is introductory, too introductory for many of you. I didn't know who my audience was going to be. But basically, the analysis of biochemical reactions is governed by the law of mass action, in which we can describe the rate of a particular reaction as the concentration or the number of particles of a particular reactant multiplied by some rate constant for a first-order reaction. And for a second-order reaction, the concentration of the two participating substrates multiplied by a second-order rate constant. And I'm going to keep coming back to, during this talk, the Michaelis-Menten system, which most of you or all of you are probably familiar with. No? Okay, maybe you're not familiar with it. Okay. So Michaelis-Menten is, a classic system in which it's very simple. You have an enzyme, we have a substrate. The substrate binds with the enzyme, forms an enzyme-substrate complex. Do we have that laser pointer? Oh. So yeah, so it forms an enzyme-substrate complex. That's basically governed by this reaction, and this has a particular rate constant, which I call K1. This complex can then dissociate back into the substrate-enzyme complex with a separate rate constant K2, or the enzyme can act on the substrate and produce the product. The enzyme goes on to re-react with other substrates. This is governed by a third rate constant, K3. So in this Michaelis-Menten system, we have three reactions that can take place. We can rearrange these reactions to create a differential equation which describes the change in concentration of substrate over time. And this is a function of the two rate constants, or two of the rate constants, and concentrations of the relative substrates. So given these three reactions, we can actually, and because there are four substrates participating in this system, we get four differential equations, one for the substrate, one for the enzyme, one for the enzyme-substrate complex, and one for the product. And we can actually solve these deterministically to show how the system changes over time. And so in this case, we would normally start with a high concentration of substrate, a relatively low concentration of enzyme, no enzyme-substrate complex, and no product. And as you can see, as the time progresses, substrate decreases to 0, product increases to some limiting factor, and in the middle, during the highest reaction speed, the enzyme-substrate complex reaches its highest concentration, and the enzyme reaches its lowest concentration. This is just a sample system, by the way, so that what I'm going to be talking about really involves many more reactions than this. We're interested in doing hundreds or even thousands of these simultaneously. The problem with this situation is that biology is not deterministic. These are just a couple of cute examples. These are fingerprints of identical twins. This is a kitten which was the clone of its mother. Clearly they're not the same. And finally, we have E. coli right here, genetically identical E. coli, subject to the identical environment, or as close as we can make it, behaving differently. And so we do not have the technological capability to model fingerprints being made or cats developing fur, but maybe we can approach the complexity of simulating what these bacteria are doing and reacting differently to to the same environment. And I should say, a lot of this stochastic behavior, as we call it, as a result of low copy numbers of molecules inside the cell, and specifically transcription factors, if you're familiar with those, frequently only exist in the cell 10, 20 copies. So we're not talking about 10 to the 23rd molecules here. So deterministic versus stochastic simulation. A deterministic model, as I've already discussed, every future state can be predicted from a set of initial conditions, rate constants, and differential equations which describe the possible reactions. All states can be determined once the initial conditions are known, hence it's deterministic. In stochastic models, we introduce elements of randomness. We can measure fluctuations in the system. And as I just said, the low copy number of certain biological molecules really result in stochastic behavior, and that's becoming more and more evident as more and more research is done into how chemical reactions occur in living beings. So how do we model a system stochastically? So this is the pure theoretical approach, the chemical master equation. If we're interested in the probability of a particular state, x at time t, where a probability is, where a state is all the possible substrates of a system at a particular concentration, then the time evolution of this system is described by this equation. The problem with this equation is we can't really solve it, particularly when the system is quite large, and certainly when we're talking about even something as relatively simple as a bacteria. There's simply too many things going on. As a result, we use instead something called a stochastic simulation algorithm, which was developed by Gillespie in 1977. And what this algorithm essentially does is sample the space, the probability space, which the chemical master equation would describe analytically. And so what the stochastic simulation essentially says is the time evolution of a system is not a continuous process, as would be suggested by an ODE. Molecule concentrations can only change by discrete amounts. You can't get fractions of an atom or fractions of a molecule. Chemical reactions occur only when two species collide. They don't just actually, floating around, things just happen. And we can calculate the probabilities of these reactions and collisions for any number of molecules in a volume. Now, it should be emphasized, because we're not going to make this assumption in a little while, that this algorithm assumes a well-mixed environment. And like I said before, all reactions are equally probable in any region of the volume in which we're simulating. And so if you're thinking about a cell with a membrane with a lot of stuff inside it, we're going to assume that reaction A can occur on one pole just as easily as the other pole or in the middle. So I'm just going to give you a simple rundown of how the stochastic simulation algorithm works before I show you how it's been implemented on the GPU. We start out with a list of reaction constants, similar to the ones which I described in Michaelis-Menten system, K1, K2, and K3, as well as an initial state describing the numbers of the n molecule types, so x1, x2, xn. We can describe then a stoichiometry matrix, which will describe how these molecule numbers change when a particular reaction occurs. And for the simple Michaelis-Menten system, we can see if this reaction were to occur, it takes one enzyme, it takes one substrate, it makes one enzyme-substrate complex. Therefore, we have -1, -1, 1 in the stoichiometry matrix. And so, in addition, we have to modify the deterministic rate constants, which are normally designated with Ks, to stochastic rate constants, which actually take into account the volume of the system. This, without going into too much detail, is done quite easily. Then we use these stochastic rate constants to calculate propensities for each reaction. A propensity then would be basically the likelihood that a reaction will occur relative to other reactions. So if there's a lot of substrate, there's a lot of enzyme and substrate, then this reaction will have a high propensity. As the amount of substrate decreases to 0, this reaction will have a very low propensity. And you can see that enzyme and substrate, that is, makes sense with how the reaction propensity is calculated. So, very simply, we calculate propensities for every single reaction in the system. Then we sum all those propensities, and this is a good use of the parallel reduction that we were just talking about when I get to that. Calculate the time to the next reaction given the total propensity of all reactions occurring, including an element of randomness. We also choose the next reaction to occur randomly. We actually update the state of the system. We update the amount of time that has elapsed, and we iterate for as long as we wish. And this essentially will give us a solution which is consistent with what would be determined analytically by the chemical master equation, except it can be implemented efficiently or relatively efficiently on a GPU. So I'm sure you're all familiar with this picture, the GPU memory model. We know that the GPU has a large amount of device memory, 4 gig max, although more in the Fermis. That accessing that device memory is relatively slow. It has a relatively small amount of shared memory, 16 KB, or 64 KB in the Fermi, which I believe can be switched between level 1 cache and shared memory, but for our purposes,
60 or 48.
Right. We can think of it as just 64 KB. It's relatively fast, and bank conflicts are certainly a concern. So the GPU implementation. The way I've just described the algorithm, parallelization within it is not obvious how that would be done. Individual Gillespie runs do not need to share data if we consider them as being cells which are independent of one another. Cells don't need to talk to each other to know how to live. Threads within a block can share the same stoichiometry matrix and a rate constant matrix within a block. And multiple Gillespie runs can be executed simultaneously once per thread. So this is sort of an embarrassingly parallel system. All we're doing is just running a lot of them all simultaneously. In that case, it's illustrated by this picture. If we were going to do it on a CPU, we would have to calculate one cell, then the next one, then the next one, then the next one. Whereas on the GPU, we can calculate many at once. And I should emphasize that we don't, when we're doing stochastic runs, unlike deterministic runs, we're not interested in one run. We're interested in thousands or hundreds of thousands, because only that will give us the range of possible solutions that might occur, as opposed to just the one answer. So implementation. This is the initial implementation that I proposed, or that I tried. In this case, we have multiple threads within a thread block. It's a little darker than I thought it would be. Can you see that? So yes.
Why don't you point and then speak at the same time so we can...
That's much better. So we have individual threads, obviously, within a thread block, each of which are calculating their own Gillespie execution. Each thread has its own state vector. It has some workspace to calculate the propensities that I talked about. It can read, each thread can also access the shared memory of the block, which stores the stoichiometry matrix and the rate constant array. And I've tried this, I'll get into details later. This would be a good example of using constant memory, at least for this implementation, because each thread is going to be doing the same reaction at the same time, so we get that broadcast mechanism that we've talked about, which is the most efficient use of constant memory. Random numbers are also generated, and at first I tried generating all the random numbers ahead of time and storing them in GPU, in the device memory, which is one way to do it. I've eventually now just computed them on the fly. Each thread does it. And then finally, the cycle or the state of each thread is written out to first shared memory and then device memory at the end of each iteration. And this pretty much iterates for as long as you want. For this particular...
Can you talk about your random number?
Yeah, so...
I think that there are some people wanting to do Monte Carlo simulations and things who would probably be very interested to know the issues involved in doing the random numbers on the GPU.
Yeah. So it is a concern. The random numbers take up about 33% of the calculation for both this simulation method and the simulation method which I'll talk about next. Obviously, if the CUDA page provides a nice implementation of the percent twister, which is the best, they say, quality random numbers. When I tried that, it is still relatively slow. What I eventually wound up using was the combined Tausworth linear congruential generator from that book, GPU Gems 3. I don't know if you're familiar with this algorithm. It gives I believe, it only repeats every 2 to the 121 times, which is pretty good. I think the percent twister repeats every 2 to the 65,000.